So now we discuss detection of apoptotic cell based on PI binding. In earlier videos, if you can't see, you have to see that video. In that video I discuss about the detection of apoptotic cell based on annexin 5 binding. Here detection of apoptotic cell based on PI binding. In case of annexin 5 binding detection, we can't discriminate a necrotic cell from apoptotic cell and a normal live cell, but so we have to perform a different detection process, so that is called PI binding. That is why we performed the process of PI binding. So in case of PI binding, so what is PI? PI is prpodium iodide. Is a type of cationic dye. It is an intercalating agent. So first point, plasma membrane of living cell exclude charged cationic dyes such as prpodium iodide. Second point, the intact membrane of living cell exclude cationic dyes such as prpodium iodide. In case of normal living cell, the plasma membrane is intact, so it cannot retain if we add prpodium iodide, the plasma membrane of intact cell cannot retain the prpodium iodide, so it easily excludes the prpodium iodide. But on the other case, necrotic cell, so third point cell, in contrast, due to their extensive membrane damage, necrotic cells are quickly stained by PI. So in case of necrotic cell, the plasma membrane already highly damaged by any type of factor like trauma, any type of toxin infection. Necrosis, first of all we have to know what is necrosis. Necrosis is a highly pro-inflammatory form of cell death, results in release of alarmins or danger signal such as heat shock protein, uric acid, ATP, DNA and nuclear protein that alert and activate the innate immune system. So it is not a programmed death. In case of apoptosis, the apoptosis is a programmed cell death, but it is not programmed. Like any type of trauma, infection or other factor, external factor, harmful factor such as toxin can lead to a cell into necrosis. So necrotic cell shows a high damage in their plasma membrane, so they can easily retain the prpodium iodide. On the other side, the apoptotic cell uptake prpodium iodide, but that is much slower than that of necrotic cell. So in brief, like normal living cell, the plasma membrane of normal living cell is intact. So the normal living cell plasma membrane excludes the charged cationic dye prpodium iodide. But on the other side, necrotic cell cannot. Necrotic cell can retain the prpodium iodide due to their highly damaged plasma membrane. Now if we want to elaborate the diagram, we can see that the box that contain the cells that contain live cells, the box of the live cell, they are annexin 5 negative and also prpodium iodide negative. Early apoptotic cell, on the other side, early apoptotic cell annexin 5 positive but prpodium iodide negative, because the plasma membrane of the early apoptotic cell does not highly damaged, so they cannot retain the prpodium iodide. In case of necrotic cell, we can see that they are both prpodium iodide positive and annexin 5 positive. Late apoptotic cell is also prpodium iodide positive and annexin 5 positive. So in case of detection of apoptotic based on PI binding, in this case prpodium iodide is a cationic dye, and the normal plasma membrane cell easily exclude the cationic dye prpodium iodide. But in case of necrotic cell or late apoptotic cell, they can retain the prpodium iodide because their plasma membrane is highly damaged. So and we can detect the process by a technique that is flow cytometry. Thank you. If you like the video, please share.